So I'm going to take a slightly different perspective on the whole thing, which is complementary, of course, which is one of data. It's thinking about how can we help accelerate biomanufacturing by considering the role of data in that. It's obviously critical and yet in some ways overlooked. And after 20 years of working in more or less in the lab and both doing and leading R&D, particularly the genetic engineering of yeast for biomanufacturing, along with folks like Jeff Levency, who's on the call today. I turned towards data because I felt that our ability to deliver new products to market is as much a function of our ability to harness data as it is the ability to move atoms around in the lab. So put that in perspective, I think it's useful to think about history. You know, we started, perhaps there's other examples, but one of the first bioproducts is glutamate. It took about 50 years to get to a manufactured process with wild type organisms. And since that in 1957, we've obviously gone a long way. In 2006, a truly revolutionary product in my mind was the DuPont Tate and Lyle work on 1,3-propane diol. That took about 15-ish years, since that was a 6-gene engineering effort. In 2011, I had the privilege of working on the farnesene development program. That took about 6 years, 22 genes engineered into that organism. So obviously the complexity is getting higher. The time to develop is getting shorter, and that's pretty exciting. It's only going to get better, but there's a kind of a cloud there too. If you look at the same from a cost perspective, it's getting more and more and more expensive. And these dollars I'm putting here are not the production costs. These are the R&D costs, the development costs to get that product to the manufacturing floor. And the problem with that is that the higher the capital investment and the greater the uncertainty in getting those products developed, the fewer and fewer products we can actually consider putting on the market. So if it costs $100 or $200 or $300 million to do the R&D, you got to have billion-dollar, multi-billion-dollar markets to generate the enthusiasm from investors to invest in that. But there's a lot of really valuable products out there that might be 20, 40, $50 million a year markets. And in aggregate, that long tail is actually a huge portion of the economy. But we cannot access it because nobody's going to invest when it takes $150 million to develop a product for a $10 million a year market. So I think there's lots of room to get there. I'm excited about the future, so it's not all doom and gloom. In fact, I take inspiration from this work. This is a plant that Jeff built with the team at Amyris in Brotas, Brazil. Those are 3 of 6 200,000 liter reactors for manufacturing farnesene from genetically engineered baker's yeast, beer yeast. And we were able to accelerate the process of taking new strains from the laboratory to these tanks in as little as 3 months. And getting to that point was no small accomplishment. It was the work of hundreds, harnessing both the advances in genetics and bioprocess technology, but also improving our data systems. And Jeff gave a brilliant talk on something like 25 lessons, a couple dozen lessons he's learned over the years, not just farnesene, but also Tate and Lyle and many other places. I can't talk about all of those. I'm going to pick out 2 of the lessons that were critical in helping us get these products to market quickly. And that's process quality and aggregated data. Those really, to me, are foundational. So here's what I mean by that. I'll give you an example. Early on in the development of those products, the genetically engineered and the bioprocesses at Amyris, we struggled with noisy data. We had processes that looked like the one on the left, where as the fermentation progressed, you get all of these data points and they were kind of a cloud and you get an average behavior. And then you take that strain, what you thought was a new, improved, better performing strain, and you throw it over the fence to manufacturing and say, look, we have the world's greatest strain, go make it happen at full scale. And what would happen, you see double-digit drops in performance, and it would take a long time because people would be troubleshooting and frustrated, and there'd be a little bit of finger pointing going on. It's your fault. No, it's your fault. And then we started to introspect and said, what are we doing wrong here? And as we looked and looked and looked, it actually was interesting. The manufacturing problems were not manufacturing problems. They were R&D problems. What we were doing was driving or throwing strains over the fence that were actually not what we thought they were, because we had so much noise in our measurement processes and our R&D processes that we were getting what were effectively artifacts of experimentation that weren't performing as we thought they were, and sending them across. So what we did is we focused on taking the methods of total quality and process engineering from what is typically done in manufacturing into the lab. In other words, we kind of scaled down the problem instead of focusing on scale up. And doing so, we actually we got the noise out of the system. We took our dirty glasses off, and we could start to see what was going on with those processes. And this black line here is a differential equation model, a full simulation of the physiology and reactor dynamics at small scale, which turned out to be predictive of large scale. And when we understood the problem, we could go from 0.5 liter to 200,000 liter, more or less, with nothing in between, and do it in 3 months and get exact performance matching across scales. So the key to scaling up was actually scaling down and cleaning up our act in R&D. So something else comes from that kind of process quality, the ability to aggregate data. And we talk a lot about machine learning and how amazing AI and machine learning are going to be, but you cannot apply machine learning to 10 data points. A typical bioprocess or a batch fermentation generates a handful of data points about a specific condition. So that might look like this, where we're testing a dozen conditions in a particular experiment. But you can't see a correlation from that. There's no patterns to recognize until you aggregate it with hundreds of experiments. And that's what happens when you put 500 high quality experiments together. Suddenly these relationships emerge. And the only reason we could put these together is because each one of those those batches were done with extremely high quality. The noise was taken out, and we could trust each and every mass balance and each and every measurement with absolute precision. Then you can aggregate it and you can learn from it. And we saw this relationship pop out between yield and a particular parameter, which we were able to use to augment a predictive model of fermentation dynamics. And out of that, accidentally, in a way, popped an observation that if you just add water to the fermentation at the beginning, you get more product out. And that was the prediction. You get a 5 to 10% production gain, essentially cost-free change. And guess what? It went all the way up to 200,000 liter scale, and it works. The human side of that is actually interesting too, because there's a really brilliant process development scientist at Amherst, Paul Hill. And I have to give him a ton of credit, because it wasn't just about making model predictions. It was about a risk-taking mentality, a controlled risk. And he's like, it's kind of crazy, but let's try it. And he tried it, and it worked. And I think that there's also a human element to this about working together in teams. And Jeff addresses that in his learnings as well, but critical for scale up. I won't go into that. I just wanted to mention that the human element is often also overlooked. So related to the human element is communication, is how do we communicate and collaborate on these processes. And along the way in my journey in R&D, an email came across where somebody was trying to communicate challenges they were having in scale up and scale down. And they drew this napkin sketch and they sent it by email. And they said, we should really get this down on paper, because this is what we're trying to do and it's not quite working. We'll get it worked out. And I thought, oh my God, this is how we're sending our quote blueprints around in the process sciences. Why don't we have CAD/CAM concepts to communicate? And because we don't, people spend months putting data together, or they change what's obvious to them, but not necessarily what matters. Jim Collins, another former BU professor, used to say, we look under the lamppost for our lights instead of walking around the parking lot in the darkness to find where we look under the lamppost for our keys rather than walking around the parking lot. And that's kind of what's happening in the process. So that became the basis for Riffin's approach to this. We're not going to solve the whole problem in biomanufacturing, but we're going to try to solve a big part of the data problem by creating blueprints for process technology, which articulate a series of actions as well as the specifications that are product and process specifications that are constantly evolving. And you can see the versioning over on the left, so that you can iterate your way to higher performing processes. And when you're ready, you just press a button and transfer those blueprints to manufacturing for immediate population into your manufacturing execution system and then operations. And we have successfully developed this system. It's in use at 7 of the top 25 biopharma companies, as well as a lot of industrial biotechs. And it not only allows you to diagram these processes, allows you to articulate data on those processes. And by doing so, you combine the context of how an experiment was done with the actual data so that we can automatically assemble experiment after experiment into those aggregates I talked about before, so that you can do machine learning in real time. And the benefit of that is that when it becomes easy to access data in machine learning ready form, you start to ask questions, not just with fancy AI, but also with statistical tools like regression analysis and leading analytical software to inspect the relationship between process variables and outcomes in a way you never did before. And that leads to profound impacts. We've been able to accelerate the time to market for a biofuels project at Novozymes by 2. So they got there faster, twice as fast as any historical development program at Novozymes by using this approach. Reduce effort by 25% in bioassays for translational medicine. Reduce data preparation by 20x for fermentation in an industrial biotech company. We were able to get a new clinical assay with Imperial College, working together with Imperial College, in 9 weeks to the clinics at the NHS for COVID, saving 16 hours per batch for solid dose formulations. And we've reduced animal use by 80% for some of our customers who work in veterinary health. So the payoff is huge from using data more effectively. And when coupled with these innovations in genetics, bioprocess, and mechanical technologies and everything else, I think we can start to achieve that prize of lowering the cost from hundreds of millions of dollars for new products to ones of millions of dollars, which then opens up a whole new world in the capital markets for innovation. So those are my thoughts. Thank you.